Okay, welcome to another week in review of Level 1 Genetics for NCEA. And the previous video, we talked about the structure of DNA and the fact that the DNA molecule was the actual entity which was passed from generation to generation and was accountable for the variation that you had. We talked about the variation within the peppered moth, and then we started to look at the actual detailed structure of DNA. So there are a couple of things I want to cover in this video. The first one we're going to look at is DNA replication. This process has quite a few steps to it, so we had fun animating that in lesson this week in an effort to try and get you guys to memorize the steps. The triplet code and proteins, this is just a little addition to the replicative process, is that if you want to get proteins, you have to go a step further. And then number 3, we looked at sexual versus asexual reproduction. The sexual reproduction in humans is what we're interested in here. And then in yesterday's lesson, we introduced the process of cell division, specifically meiosis. Mitosis is briefly mentioned in level 1, but meiosis is the one that you need to be more familiar with. Okay, so DNA replication. There's a couple of steps to DNA replication. The first thing is you need to be aware of where these processes are taking place again. So we're talking about DNA replication first of all here. In the last video, I helped show you guys the link between genes, DNA, and chromosomes. So DNA replication, everything is going to originate inside the nucleus of a cell. So that's the nucleus right there. This process can take place in any cell which has a nucleus and DNA inside that nucleus. So basically, almost every body cell. So you have the DNA molecule in here, which is wrapped around a chromosome. So that's a chromosome. And then the DNA molecule, we just put that in red. It's wound around the chromosome like that. So what we're focusing on now is copying this DNA molecule. So we're just going to zoom out a bit here. And now we have our particular DNA molecule, which is wound in its double helix pattern. You want to copy this DNA molecule. There's a couple of reasons why. If you want to, number 1, make some new proteins for the body. Now, proteins make things like enzymes, hormones, cell receptors, etc. There are many different functions of proteins in the body. So this process is constantly happening inside the body, and therefore you must be able to understand the replicative process in order to make proteins. So with our DNA molecule, I'm just going to focus on 4 bases. So G pairs with C, we did that the last day, and T base pairs with A for correct base pairing. And the first step in DNA replication is that you have to unwind that double helix. So if you just go back up for a second. So this double helix has to be unwound. So the first step, step 1, is unwinding of the double helix. Okay, so if you unwind that double helix, at the bottom of it, it's going to look like this. So it's nice and straight now. And then we have G and C still base pairing, so there's still base pairing there. Then you have other bases as well, which are still base pairing as well. So we've unwound the bottom section of the DNA molecule. Now the next step is to unzip it. So step 2 is unzipping. And the idea there is that the DNA molecule, when it's straight, it looks like a zip there, and you can see the teeth. And the unzipping means that it just needs to separate in each of these directions here. So when it unzips, it's going to be a bit more open. So let's draw it like this. Still not the entire helix now. This happens and it works its way up the length of the molecule. So the bottom section now has been unzipped. The A and T have been separated. The G and C have been separated. And then the other bases just starting to separate as that unzipping moves its way up the length of the molecule. And once the molecule has been unzipped, the third step can take place, which is to add new nucleotides. Remember, a nucleotide consists of a sugar, which has 5 sides to it, so that's your sugar, and then a phosphate, which is stuck on the side there, and a base, where the base can be G, T, C, or A. So you guys, we covered that well in lesson and in the last video. So we're adding lots of new nucleotides. So what I'm going to do now is use a different color for the new nucleotides. So in the situation we have here, if the molecule has been, so we're unzipping the molecule still, so let's continue like this. We have T here, G there, and all the other bases. And we have A here and C here, and all the other bases continuing up the molecule. Now with the new bases, the new nucleotides, let's use a green color. So we're going to add these new nucleotides in green. So we'll have A with T there, and we'll have C with G, and then other bases will start to add themselves. And then these, of course, are nucleotides, so they're all linked together as well. T with A, G with C, and so on up the length of the molecule. So we're starting to add new nucleotides to the original DNA template, the red one. And then the final step will be to end up with two new DNA molecules. So two new DNA molecules, so it's going to look like this now. So if we just go from here to here, we're going to have the original DNA strand, which was in red, and just continues with bases up along the length of it. Another one of the original DNA molecules, but the first base in that case was A, the next one was C, and then continued up the length of the molecule. And then our new strand, which was added in green. So get the correct green for that. This green right here was A, C, and up the length of the molecule. That ends up being in green. And then we have the same sort of situation on the other side, T with A, G with C, and then the molecule continues up the length right there. So this process of DNA replication is sometimes described as being semi-conservative, because what you do is you conserve the old strand, that's the old DNA strand in red, which is referred to also as the template strand because that's the one you build upon, and then the new strand in green. So it's half new, half old. That's why it's described as being semi-conservative. In any case, the outcome of DNA replication is that you have replicated the original DNA molecules. You've gone from one molecule to two DNA molecules, and they are identical to each other. So once this DNA is being doubled up and replicated, the next question really is, well, what are you going to do with this new extra DNA? So the first thing we can do is we can use it to make new protein. And this is a process of translation is what it's known as. Translation. Okay, so to make new protein, we need to talk about the triplet code. And for level 1, basically all you guys need to understand is that out of the nucleus within the cell, some of that DNA molecule comes out through the nucleus. And then you have your bases reading along the length of that. No particular order in this case, I'm just writing down a random sequence of bases. And as those bases feed out from the nucleus, certain enzymes will come along and they will read 3 bases at a time, and 3 bases will code for 1 amino acid. So if I use little shapes for the represent amino acids, I'll use a square to represent 1 shape, and then you read another 3 bases, and that will introduce another shape, say, let's say a circle, and then those amino acids start to stick together. And a chain of amino acids, so this is a chain of amino acids. So a chain of amino acids is known as a protein. And then you can make your protein molecules for enzymes and hormones and receptors and all those things that the body needs. So it's called the triplet code because you need 3 bases. 3 bases code for 1 amino acid. That's why it's called the triplet code, and that is enough detail for you guys for level 1. That is how you form proteins from DNA, and as I say, that can be necessary for various processes in the body. Now, the other reason you would need to double up the DNA content, which tends to be assessed more frequently in your exams, is for cell division. So I'm going to take a new page now, and I'm going to start talking about cell division. Okay, so number 2 was supposed to be triplet code, so we've done that. Triplet code was just a brief little discussion there. So we've done that. Third thing I want to talk about then is reproduction. And you have 2 types of reproduction. You have sexual reproduction, and then you have asexual reproduction. And just briefly to talk about the advantages and disadvantages. Advantages of sexual reproduction. The main advantage is that you have introduced variation, lots of variation from one generation to the next. Whereas with asexual reproduction, you're essentially cloning yourself, cloning process, and therefore there's no major variation from generation to generation. This happens in bacteria. We talked about how a bacterial cell, there's no nucleus actually around the DNA in a bacterium. So all it does is it goes through a process called budding, where it just forms a little side part to its cell, and then eventually it gets bigger, and then it becomes two separate cells. And these two separate cells will be clones of each other if the process goes without any damages. So the disadvantage of asexual reproduction is that there's no major source of variation. The advantage, though, is that it's fast. You don't need, you don't need two partners to make it happen. It's fast, and also they tend to be able to divide very, very quickly. Sexual reproduction introduces variation from one generation to the next, which is why it's really important for humans. It's a slow process in terms of the time it takes for the baby to develop, and also you have low numbers. So bacteria can be producing thousands and thousands of themselves within 20 minutes, whereas we take 9 months just for one child, or sometimes two. Okay, so they're the major advantages for and against sexual reproduction. So with human genetics for level 1, we are focusing on sexual reproduction, and a particular type of cell division which takes place is called meiosis. So let's get on to meiosis now, and we'll take a look at this on another slide. So a new page, we're going to look at the process of meiosis. Now I'll just briefly compare meiosis and mitosis for you guys so that you understand the difference. Okay, so meiosis is first of all for the production of gametes. So production of gametes, also known as sperm and egg. Okay, so meiosis is for the production of the gametes, the sperm and the egg. Mitosis is just normal cell division. So say you want to grow more skin cells or hair cells, then they'll undergo this process of mitosis. Meiosis, you have one cell, best described with a diagram really. So from one cell, which would have had the normal number of chromosomes, 46, which we described during the week as being the diploid state. From that one parent cell, you will end up, first of all, you double the content and you make 92 chromosomes through the process of replication, DNA replication, which we just described. And then that will further divide into 4 daughter cells, and these 4 daughter cells will have 23 chromosomes. So 23 chromosomes in each of those, and this is the haploid state. So the gametes here, this could be 4 sperm cells or 4 egg cells if it was in the ovaries of a female. So meiosis, you have one cell from 1 to 4. So from one parent cell, you end up with 4. Whereas with mitosis, you start off with the parent cell, 46 chromosomes, diploid state, and then you double up the content through replication, same as before, and you end up with a cell with 92 chromosomes, and then that divides into just 2 cells, and those two new cells, will have 46 and 46 chromosomes. So you're forming diploid daughter cells, and this is very like replicate or cell division in bacteria, because from one cell you get 2 cells with the same DNA content, and this could be two new skin cells. Okay, so that's the main difference between mitosis and meiosis. So from meiosis, you get from one cell you get 4 mitosis. From one cell you get just 2 new cells. And then in class I said just remember T for 2. So therefore you can't mix up mitosis and meiosis. The detailed process of meiosis we covered in class, because you guys need to understand the movement of the chromosomes during that process. So what we're going to do is, if I redraw the process for meiosis and then put the detailed cell division steps beside it, that should help. So meiosis in detail. The overview again was from one parent cell with 46 chromosomes. So this could be in the testes of the male or the ovaries of a female. And then from that, as I say, it doubles up its DNA content through replication to have 92 chromosomes, and then it divides to give you your 4 cells with 23 chromosomes in it. Okay, so what we're going to do now is we're going to look at this process in a bit more detail. We're only going to consider 4 chromosomes because otherwise it's a bit too complicated to draw 46 every single time. So this is the cell, and what we're going to do is we're going to expand the nucleus now to pretty much fill up the majority of the space of the cell. I'll use blue color for 2 large chromosomes, and then I'll use a different color for 2 shorter ones. Let's use this green right here. So you have 2 large chromosomes and you have 2 small chromosomes. Now, the first step then is to undergo this replication process right here, so DNA replication. Now, with the replicative process, what we're going to end up having is we're going to end up having the chromosomes essentially growing a second branch on itself. So go back to my blue here. So the large chromosomes here, they will form 2 legs now. So they've doubled themselves up. And then the little short green ones will do the same thing as well. They've doubled themselves up. And that was the first round of replication. So you've gone from a situation where you had your 46 chromosomes, that would be the 46 situation, now to your 92 situation. Okay, so hopefully you guys follow that so far. Then the next step is this particular cell here needs to actually divide itself. And how it's going to do that is it's going to form a bump like this, and the chromosomes are going to line themselves up. The small ones will line up, they'll pair up together. Now I didn't do that very well. I should have had different colors for each of the big ones and the small ones. So let's just change that right now. Let's make that one a large orange one, and then make this one a small orange one, and make this one a small orange one, and a small orange one. Okay, so the point of that is that the homologous pairs, so homologous, which is the same in genetics, that means the same. Homologous pairs match up. So basically the small chromosomes come together like this. And what we want to do is we want to bring them a bit closer so they're actually in contact. So I'll use my orange for that. We'll bring them in contact with each other. And then we'll do the same with the blue ones. So we had a large blue one and a large orange one. So large orange and large blue in contact right there. Okay, now this process we're trying to illustrate right here in this diagram is very important. This process here is called crossing over, and we just zoom in a little bit. The large blue chromosome is crossing over, in this case, with the large orange one, and they can do it at both ends. So you got crossing over right there and crossing over right there. Now crossing over allows for another little bit of variation to be introduced into the chromosome, and you'll see that the result of that crossing over when we fully divide these 2 cells. So from that one cell with 92 chromosomes, we now have 2 cells which effectively would have 46 and 46. The small green chromosome is over in one side of the cell, so that the cell has been split this way and this way, and we now have 2 separate cells. So we have a green one on this side, and we have the blue one has also been pulled. The blue one has been pulled to this side of the cell as well. The orange and the 2 orange chromosomes have ended up both in the same cell. That's just the way this particular replicative process has happened. So whichever side the chromosomes end up is called random assortment. A bit of the orange has exchanged with the blue, so there's evidence of that on that chromosome, and the little orange one has also exchanged with the green, in which case there should be evidence of that on the other chromosomes. So from crossing over, there's a bit of the green on that one, and then there should be a bit of the blue gene onto the large orange chromosome, which you can see right there. And that's as a result of the crossing over process. So that introduces some variation with crossing over. You get some additional variation right there. And then, of course, we still have 46 chromosomes in each of the cells. So we're not ready to form gametes just yet. So it undergoes a further division process. And we'll just go back to our green. You'll end up with your 4 cells all together. And in that, what happens then is the chromosomes themselves, which have originally been doubled up there, they'll separate out now into a situation where you have the pieces of them left over. And we want the orange there, so there was an orange piece. That's in the wrong cell. You can see that hopefully yourselves. That orange piece should be in to this cell, so I'm just supposed to put it on the top of that there. That's what I was trying to do. So that would be that section of it has gone there, and the other blue part has gone in here. The little green one then has been moved there, one part of it, and then the other part, which had the orange section on the top, is in that cell. So the colors, although this is a bit tedious on my part, it really does show the fact that you have nice variation of colors, and therefore variation of the type of children that you will get from this. So you have a bit of blue and a bit of orange, which would represent that section right there. We would have a complete orange chromosome right there. Also a small orange chromosome, and over here a small chromosome with a bit of green. Small orange chromosome with a bit of green, which is from that section right there. So that is how you get assortment and variation within the meiosis, the process of meiosis. So it gets quite tricky. I've never seen any exam paper ask you guys to describe those steps that I've just done. You can see it was quite tricky even for me to do. But you do need to understand the fact that you can have variation through crossing over, that you can have variation just by how the chromosomes assort themselves into the different cells at the end of the process. And then, of course, if these were 4 sperm cells, then the sperm cell itself would then have to join with an egg cell, which could have totally different colored chromosomes in that. And then you get even more variation when those 2 nuclei combine and form a zygote, which will develop into a child. So there's lots of variation through the process of meiosis and then through the fertilization process itself. And this accounts for the variation within human beings during sexual reproduction. Because this process doesn't happen in bacteria, there's very little variation within bacteria. So that was DNA replication. We also did the triplet code. We talked about the merits of sexual reproduction versus asexual, and then I talked about meiosis and mitosis, and finished there with a detailed process of meiosis itself. Next week, we will be looking at how these different genotypes present themselves in phenotypes. So these different combinations of alleles, they will actually appear in an organism. And we just briefly mentioned Punnett squares. So that's what we'll be looking at next week.